I call upon to the dais the chairpersons for the next session, Dr. Chandrasekhara Meshram, Dr. Gagandeep Singh, and Dr. Satish Chandra. I request the chairpersons to start the session.
Yeah, good morning. We are here for a very interesting and prestigious Professor N H Wadia Oration. The first N H Wadia Oration was held in 2004 and it was delivered by Professor Louis Kaplan in Nagpur. And today we are having this oration. Professor N H Wadia is lifetime professor at JJ Hospitals and Government Medical College, Grand Medical College, Mumbai. He had a lot of research experience, and the notable research was SCAD2 and then polio-like illness because of enteroviruses. He established the neurosciences department and he was director at the Jaslok Hospital, Mumbai. And he has various important awards, and the most important award was Padma Bhushan by Government of India, and he was Vice President of the World Federation of Neurology, and he was received the prestigious gold medal for services to international neurology. So, ladies and gentlemen, welcome to this Professor N H Wadia Oration. Thank you. Who is production? The oration will be delivered by Professor Imad Najm, and Professor Ram will introduce the orator. Thank you.
Good morning, everybody. It's an absolute honor to introduce Dr. Najm, and I would like to thank the organizers for IAN for giving me this opportunity to introduce one of my mentors. Dr. Imad Najm, he's the director of Cleveland Clinic Epilepsy Center in Cleveland, Ohio, and he's the vice chair for the Neurological Institute there. He received his medical degree from St. Joseph University in Beirut, Lebanon. After finishing an internship in Paris, he became a postdoctoral fellow in the Department of Neurobiology at the University of Southern California. And during that time, Dr. Najm's research was focused in the basic mechanisms of status epilepticus and neuronal injury. He finished his residency in neurology at Cleveland Clinic, followed by a fellowship in clinical neurophysiology and epilepsy. And he is board certified in adult neurology from American Board of Psychiatry and Neurology, and also in clinical neurophysiology and epilepsy. Between 2000 and 2003, Dr. Najm served as the director for Epilepsy and Clinical Neurophysiology Fellowship Program at Cleveland Clinic, and he later became the head of the section of adult epilepsy. And since 2006, he's assumed his current position as the director for Cleveland Clinic Epilepsy Center. This is one of the largest epilepsy centers in the United States, and it accounts for about 2.2% of all epilepsy and seizure diagnosis in the country. Over the last 22 years of Dr. Najm's experience in clinical and basic mechanism of epilepsy includes numerous research projects dedicated to exploring the mechanisms of epilepsy and malformations of cortical dysplasia. He has authored more than 300 research papers, and his citations are north of 23,000 at this time, including some high index citations. He's authored or co-authored more than 17 book chapters in books. He's an excellent artist, and he's good at drawing neuroanatomy illustrations. By far, I think the greatest legacy for Dr. Najm, he's an internationally recognized expert in the world of epilepsy surgery, and he's instrumental in bringing the stereo EEG technique not only to North America and US, and because of his work, I think stereo EEG is being used as widely nowadays, not just in America, but through his students and fellows in most of the countries in the world. I can personally know some of the fellows that he has trained, not just in fellowship, but personally made sure their centers are also successful, including in India and several other countries, which are now routinely using stereo EEG. And thank you, Dr. Najm, for changing the world of epilepsy surgery. If this was not enough in him changing how we evaluate and treat epilepsy patients, I think his next projects are even more important, which have the potential to fundamentally change our understanding of neurological disorders and how we treat them in future. In January of 2022, Dr. Najm launched an ambitious project called Cleveland Clinic Brain Study. It's a one-of-a-kind, large-scale longitudinal study, which is aimed to observe the early underlying changes of neurological disorders. The aim is to recruit about 200,000 or 2 lakh patients with follow-ups for up to 20 years, and the goal is to uncover the pre-symptomatic causes of neurodegenerative diseases with yearly brain scans, echoes, biomarkers, and eye imaging. I think this will give us insights into why things happen, detect them early, and get the treatment started early on. I can go on and on like this. His work is important in making Cleveland Clinic one of the ranked as the #1 smart hospital in the world, which includes projects like the first quantum computer dedicated to healthcare. Thank you, Dr. Najm, for changing the world and making it a better place. And without much further delay, I would like to hand it over to Dr. Najm. Thank you.
Professor Imad Najm, the stage is yours. Thank you. Thank you for being with us. He had been a victim of a bomb hoax, and so he could not reach the first day. Thank you for being with us. Thank you.
Thank you, Mr. Chairman. Thank you all for the kind invitation. Such a big honor for me to be talking in front of all of you, talking to giants in the field of neurology and epilepsy here. So I'm humbled by this, and I hope I would not disappoint here. And thank you, Ram, for this very kind introduction. I know you're biased, but really appreciate it. And last thanks is to my good friend, Dr. Sanjay Singh, to be encouraging me here and to be a big part of the invitation for me. So 2024, it marks the 100-year anniversary of the EEG. And I thought quite a bit about what should I be talking to such a very distinguished audience about. And I chose this talk to reflect a little bit about the field of epilepsy and epilepsy surgery in particular, 100 years after the EEG was discovered or was first used. So I thought in this next 50 minutes or so, I will be talking a little bit about epilepsy before, how it was, and then the impact on EEG and epilepsy surgery in particular. Then reflect on the current challenges that are facing us in the field epilepsy nowadays, then try to put some thoughts to how we have a pen here to chart the next 15, 20 years of epilepsy care. How would it look like? So Hans Berger in Vienna, Germany in 1924 recorded the first brain waves. And you see here in the bottom of this slide, you see the first recording that is later to be known here as the alpha waves or Berger waves. And then it enabled, first time, all of us had to see how this brain is functioning from the electrophysiological. And then, since then, we all know how often we've been using EEG in the context of evaluation for epilepsy surgery and epilepsy in general. So, did epilepsy look like in the hundreds of years before the EEG recording in 1994? So epilepsy was often associated with supernatural causes. It was a curse, or thought to be a curse, a possession, or a divine punishment in ancient cultures. It was misunderstood up to the even remaining 1950s, 60s, 70s, as to be a form of insanity, as a mental health disorder. Yesterday, when Dr. Chowdhury was given his presidential talk, so many similarities, the history of headache, migraine headache in particular, and epilepsy, the way they were both perceived. And patients with epilepsy, they were stigmatized. Unfortunately, this thing remains one way or another in our society and probably societies all over the world. And there is this unconscious stigma, unconscious bias that does remain. And when we look here about epilepsy as a medical condition and what did we know about it, there was no tool or no methodology besides clinical history and exam to confirm the diagnosis of epilepsy. And the mechanisms of epilepsy were not known. There were only a couple non-scientific, with current day definition of scientific hypotheses that were advanced. Then, in the late 1900s, all the way to the earliest 2 or 3 decades of 20th century, there was a transition from the epilepsy surgery standpoint from trepanation as to basically relieve the brain, the pressure on the brain, and get the demons out that were possessing patients with epilepsy. And from rudimentary type of treatment that relied on superstition, isolation, or herbal type of treatment with no scientific evidence. But there was some bright development in 1857 with the identification of bromide as potentially the first medicinal drug used. And then in the earlier years of the 20th century, there were identification of phenobarbital as another medication. Epilepsy surgery was nascent. So here, these are some of the events in particular that may be important or made a significant impact and continue to impact our thinking on epilepsy surgery. there has been an increasingly critical view of trepanation in the late 19th century and the science of phrenology. And then, because of work of people like Broca in France, Perrier in London, England, Hitzig and Fritsch in Germany, and their stimulation studies on animals, there was now new development about localization of function that is specific of areas, in lobar areas in the brain. And then, last but not least, there was the first attempt to define what may be leading to the generation of epileptic seizures by Ewing-Jackson. So Ewing-Jackson talked during an oration at the Harveian Society in 1879, London, England, about the concept of what he called the discharging lesion. For the first time, somebody advanced something related to electricity happening in the brain, these neurons. And he talked here, part of his oration, about, I'm going to read here, highly unstable region of the cerebral cortex. Nothing more than a group of cells, meaning neurons here, whose instability is raised above normal. Very, very accurate description now, almost 150 years later, about the epileptic lesion. But they had no ways to verify or validate this hypothetical definition of the possible cause or generator of epileptic. This led the group, this work on better identification of the function in the brain through direct electrical stimulation by the groups of Broca, Fritzig, and Ferrier in England. The group that consisted of Ewing-Jackson, Ferrier, and Victor Horsley, the neurosurgeon, at the hospital for the paralyzed and epileptics in London, that now is known as Queen Square in London, England, to start to treat patients with difficult to control seizures. And the first 3 cases they operated on were patients with depressed skull fractures in the area affecting the primary motor cortex. And these patients had focal motor seizures and sometime evolving into taking half of the body than before secondary generalization, what we know as the Jacksonian march of these focal motor. And the hypothesis is, if these patients are having these focal movements on one side, there should be the epilepsy or the discharging lesion should be in the contralateral side in the primary motor cortex. So the concept here that there should be a lesion and there is a depressed skull fracture. And when this happened here, they took these patients to the operating room and they did direct electrical stimulation to reproduce at least part of the seizure and to do the resection. And within 2-3 years, the group did 40 cases. Only 10 of them died, which was a major, major achievement, knowing that the neurosurgery in general was not safe because of the no antibiotics, infection, and major problem with anesthesia. But their work laid the foundation that persists till now in thinking about how we do epilepsy surgery and how the concept of lesional epilepsy that the North American and the English schools continue to use extensively up to the better or more utilization of SEEG. Now, EEG was recorded in 1924. It enabled, as we all know, enabled us to differentiate between types of epilepsy, confirm diagnosis of epilepsy, and was or became the primary biomarker for epilepsy, for good or for bad. And actually, it is the only biomarker that we have to confirm diagnosis of epilepsy even in 2024. And the technology here evolved with the years. You know, technology of recording went from analog recorders to the current now much more sophisticated portable type of recording that are digital recording with digital amplifiers. Now, if we look here at some defining events in the history of epilepsy surgery before and after EEG, of course, we have to remember the work of Ewing Jackson, which hypothetical works that laid the foundation of the discharging lesion. And then the work with Victor Horsley and Jean Ferrier in London to start epilepsy surgery based on lesion. And then we have the work of Förster in Germany after the EEG to start to do mapping of the human brain and defining what later to be known by Wilder-Penfield, through Wilder-Penfield work as the homunculus, the homuncular representation of the function in the brain. And Wilder-Penfield at the Montreal Neurological Institute took the surgery to more modern approach. And the biggest contribution in the field was direct electrical recording in the operating theater from the lesion, surrounding areas, and define the epileptogenicity and do the resection in a tailored manner that will take into account not only the lesion, but the epileptic areas as recorded intraoperatively. Almost in parallel, in the early second half of the 20th century, 1952 to 1957, the group that was led by Jean Tellerac, who was a functional stereotactic neurosurgeon, and Jean Banco, who was an epileptologist, they started approaching epilepsy in a completely different way, which is what we know now as SEEG, or stereo electroencephalography. This technology, or this methodology, was made possible by the stereotactic frames that were developed almost at the same time in Paris, France, and in Philadelphia in the United States in the 1940s. And the main contribution of this is to use not the lesion as the primary guiding principle for implanting a patient or working up epilepsy, but using what they call electroclinical correlation. Recording an EEG and analyzing the whole sequence of events in the seizure in order to map the whole network in the brain, epileptic network. And then, in 1977, Edward Damadian in New York, he basically described the first principle of magnetic resonance imaging. And this enabled us to see now the lesion for the first time in medical history, seizure lesion in the brain. And my mentor, Hans Luders, in 1981 at the Cleveland Clinic, took this work, the work of Wilder-Penfield, doing intraoperative recording and mapping, to the extraoperative setting through the subdural grid methodology. This is here the work of Wilder-Penfield at the Montreal Neurological Institute. See the lesion, touch the lesion, record from the lesion, resect the lesion and the epileptic area around it. Very simple, all intraoperative setting, one single setting. the work of Jean Banco, Jean Talleyrand at Sainte Anne Hospital in Paris. Basically, they looked, as I mentioned, electroclinical correlation to do the mapping of not the seizure onset only, because you cannot map the seizure by SEEG simply by saying this is the area where seizure starts. It has to be verified, validated by the early organization of the seizure, and it's spread later on. And here, this is the subdural grids. The main thing about subdural grid and the main basis and the strength for subdural grid was identifying the lesion, and MRI made this possible in the 1980s in the United States and the rest of the world. On the left-hand side here, you see the first MRI recording in 1977, and a report here in the New York Times titled Cancer Found Electronically. The first MRI recording was on the chest. And later, we started to see these images, the first images of the MRI, that look very much to a very low quality CT scan now. And on the right-hand side, you see this almost 40 years later, MRI imaging here using 7 Tesla magnet human recording, enabling identification of lesion as tiny as this bottom sulcus dysplasia here to enable a very, very small focal resection and successful epilepsy surgery. MRI, as we all know here, made it very possible for us to see multiple lesions, small or large, and focal or lateralized to one hemisphere. And then in the 1980s, there was in particular work by the group at UCLA in Los Angeles, California, about PET scan in the evaluation of patients with focal epilepsy. And the principle was looking a fake sugar, deoxyglucose, tagged to fluoro-18, a radioactive compound, to image the brains and look for cold spots. With the idea, if there is a lesional area, you see it less functional and help in the localization of epilepsy. Now, we know very well if we see outside the temporal lobe a very focal area of hypometabolism is very, very suggestive of a type 2 dysplasia, because there is almost complete absence of the glucose transporter in the endothelial cells in the blood-brain barrier. Now, ictal SPECT was basically socialized quite a bit by the group at the Mayo Clinic, in particular using subtraction imaging, a technology called SysCom. And in SysCom, the initial description, you see one single spot in the brain where the seizure started. And later on, we got to know that when we do an ictal SPECT, there are multiple variables, including how fast into the seizure we are doing this, and then how fast we are getting the injection. But we started to see that multiple areas in the brain are involved. And then when we started to use more SEEG, we realized actually that the ictal SPECT in many situations, in particular in patients with MRI negative epilepsy, maps most of the epileptic network in the brain, and it's the same, it's an anatomical march of the seizure in the brain that is visualized. Magnetoencephalography was a very fancy research technology used in the early 2000s in research labs. And then in late 2007, 2008, we got our first magnetoencephalogram. And under leadership of Rick Burgess at the Cleveland Clinic, we started using this as a clinical tool. It does help. It does help in MRI negative epilepsy. It does help in identifying the generator, but sometimes we give us false localization related to the spread of the electrical activity. Now, we got to learn over the years that EEG is good, but we have some issues with EEG. If we don't record a spike on the EEG, we cannot rule out diagnosis of epilepsy. That is a major weakness. The second weakness of EEG in the process of evaluating patient for epilepsy surgery is that this EEG may be mislocalizing the lobar level. It covers an area almost to be 10 square centimeter. It's big, so it's not precise when we do scalp EEG recording. And then in cases where we have parietal lobe epilepsies, for example, it may be very mislocalizing. And then we know the best way to take advantage of the EEG is to record patients for days. And could be very expensive and technologically very demanding. Now we know we're doing more EEG recordings at home, more portable recordings, but still we have issues with the sensitivity of the EEG, in particular from the spatial standpoint. Now, over the last 15, 20 years in the United States and many areas in the world, there have been some better or bigger development. And that included basically innovation in laser therapy. So we are using more laser therapy in ablating some of these small foci. It was approved in the United States in 2007. And then in 2013, neuromodulation, as illustrated by responsive neurostimulation, was introduced, followed by deep brain stimulation approval in 2018. And more recently, in 2019, high-intensity focus ultrasound was approved for essential tremor ablation and is being used now off-label for some epilepsy applications. Now, what about here the current state of epilepsy in 2024? We have in the United States more than 44 anti-seizure medications approved. 28 of them are still in use in the United States. And we all know, if we look at their method of action, mechanism of action, they target glutamate receptors or some channels, sodium channels. They target some glutamate receptors and some GABA receptors. And then potassium channels, 2 of them, but they are not used anymore. And then we do have some new mechanisms such as levetiracetam. The problem for those is none of them targets a known biomarker of epilepsy. We are targeting receptors that for the most part, they are responsible of the normal function. And that's why we always have to use this balance. We need to stop the seizures, but we need to preserve some function for the patient. That is a big problem. In 1985, this is a work by Richard Mattson and a group at Yale University, published in New England Journal of Medicine, using 4 anti-seizure medication available. 47% of the patients were shown to become seizure-free. In 2000, using here many more medication, almost 17, we have more 17 medications available. Patrick Kwan and Martin Brodie in Scotland, they basically showed exactly 47% of the patient becoming seizure-free first year after the new diagnosis of epilepsy. In 2018, a report by the CDC in the United States showed that adult patients with epilepsy, 90% of them were taking anti-seizure medications. 56% of them continue to have seizures, like 44% seizure control. So it is a big problem. So all of these medications over the years, they made some improvement probably in the side effect profile. We have not changed last 40 years. 3% of patients who are seizure-free after the first anti-seizure medications. And there may be some reasons here for this. The biggest one, we really do not have a specific biomarker, specific target that drug companies can develop medications for. We do not have, for the most part, animal models to replicate 100% the epileptic condition. And always I'm reminded of a statement, anonymous statement or a quotation that Pete Angle put it in his book in 1989 about epilepsy, saying the best model of a cat, and preferably the same cat. We don't have good models in epilepsy. It's still pertinent now, 35 years later. And we really, really suffer, like many neurological disorders, of the absence of biomarkers to use for clinical trials and for target of medications. Now we have in our possession multiple pre-surgical evaluation tools, and I basically reviewed some of them. So how we are doing from epilepsy surgery? Here I just put some samples here about it. The seizure freedom after epilepsy surgery, if we pulled many of this data, is anywhere between 50 and 80% seizure-free in temporal lobe epilepsy. And in extratemporal lobe epilepsy, it's a little bit less, 30 to 50%. That means almost half of our patients with epilepsy surgery, they continue to have seizures. Is it enough? Probably it's not enough. This is here when we looked at first almost 1,600 epilepsy surgery at Cleveland Clinic. We have, at 12 years, we have almost 42% remaining. This is not good, because most of the reports are 1 to 2 years after the epilepsy surgery. This is not enough. We cannot settle for this. So this leads us to ask this question: why do we succeed? But as important, are we still failing? So here we'll put some thoughts on this. This is a paper by the group of John Duncan in London. Basically, they talked as meta-analysis looking the data from published studies about outcome. And they looked at some good prognosticator, such as, for example, febrile convulsions history, better outcomes. Hippocampal cirrhosis, better outcomes. Complete lesion excision, more favorable results. EEG, MRI concordance, improved outcome. Severe learning disability, denoting probably bigger encephalopathic condition, they don't do very well. And they talked here that, overall seizure freedom rates, they could be good in patients with complete excision of the lesion if identified. Temporal lobe resections have been always better than others. And then we have some new conditions that up to 25, 30 years ago, we're still dabbling with in surgery, such as focal cortic dysplasia, before we advanced the classification in 2004. They do quite well. This is here a type of critical review that we published with our colleagues in Porto Alegre, Brazil, André Palmini and Eliseo Paglioli, about why we fail and how do we fail, and which is something that most of us now and many studies showing almost the same. Early on, there is a steep decline in survival, it means in patients who are seizure-free. And that initial slope, almost 50% of the failures happen the first 6 to 12 months after epilepsy surgery. And after that, there is a lower slope of continuous failures over the years at a slower pace. If we look at distinguishing features between the two, those who fail early are those patients who continue to have same type of seizure semiology, same type of frequency, and they would not respond to anti-seizure medication. These patients typically are those patients with incomplete lesional resection, or those patients who we have bad localization of epilepsy, we missed it. And since then, we should add that one of these factors now is potentially genetic factors, genetic mutation. Those who fail late, after 1 year, they tend to have new seizure semiology, much more rare seizures, and they tend to respond much better to anti-seizure medication. It's almost like a new epilepsy, where almost 2/3 of them, or 50 or 60% of them, will become seizure-free with adjustment of the medication. Why has this happened? We presume that in these patients, there is development of new focus for epilepsy in our setting. Now here, let's talk a little bit about some of these conditions. This is type 2B dysplasia. On the left-hand side, you see the photomicrograph from the initial description of focal cortex dysplasia by Taylor et al. In 1971. And what he described is these disorganized cortex with very dysmorphic neurons and balloon cells. And there is always a gradient, normal here, abnormal here, and in between, there is an abnormal piece of tissue with dysmorphic neurons, no balloon cells. Nobody made much of it till we started recording from them. If we look at the MRI of a lesion like this, for example, you have this sign, this increased signal focal. This is the nidus of the lesion. Nidus of the lesion here, you see balloon cells. The problem with this, there is no function, and there is only slowing on EEG. There is no epileptic activity for the most part here. And the focus usually is surrounding that area of the MRI, where we see dysmorphic neurons, no balloon cells, and if it is an eloquent area, this area may be functional. This is a problem to map the resection. So, if we have a complete resection of the MRI lesion without doing the recording in these cases, we will have surgical failure, and these patients will fail immediately and will have the same type of seizure. And we showed that in situation where the lesion is close to the motor cortex, if we are shying away from taking a functional area, these patients end up having status epilepticus after, suggesting, and we have some biological evidence for this, that balloon cells may be protective against a seizure. If we take only the balloon cell region, surrounding it the dysmorphic neurons, this is where we have the function. This is where we have the epileptogenicity. Now, later failures, we have some hypotheses. We have select number of cases where we have resection, patient seizure-free, and this condition for 3 years. Seizures came back a little bit different, and we found the same type of abnormality in the tissue, the FCD, focal cortex dysplasia, and surrounding it, in addition, there is a new synapse formation, exactly like the previous surgery, suggesting that we left behind a pro-epileptic lesion, but was not firing at the time of the 10 days or 15 days of intracranial recording, telling us, suggesting one weakness of EEG. It's good when a lesion is firing, but it's not good in identifying the whole extent of what we call a pro-epileptic lesion that will become epileptic 1, 2, 3, 5, 10 years later and develop a new epilepsy from it. Now, what about genetics? This is a meta-analysis study by Kees Braan from Netherlands, talking about looking at epilepsy surgery outcome and correlating this with germline mutation. What they found is very interesting. We need to confirm this. Patients who do very well are those with mTOR pathway mutations, mechanistic target of rapamycin. They do quite well. Those who don't do well are those patients for channelopathies. So that is, I think, the beginning to suggest that gene sequencing or gene studies before may be quite helpful. A couple words here about neuromodulation. We talked about the techniques approved in the United States and most of the world here. And they do well, but they do not have a significant percent of patients who become seizure-free. And one curious thing about them we're discovering, 6, 7, 10 years later, it seemed like responsive neurostimulation, they continue to get better by percent of seizure decrease, but not by the percent of patients who have seizure freedom, suggesting something like electrical stimulation is doing in that brain. So what about the future? I think our goal for the future was defined by all of us, but in a statement in 2000 by the National Institute of Health in the United States, no seizures, no side effects. I should add here, should be no epilepsy, or prevention of epilepsy should be for this. So there are steps to be taken that I will discuss for the next 5 minutes here, is basically about what we can do to, in the future, in the near future. The first thing we can do now, and we've been doing all over the world, is to see better the lesion, better imaging technologies. And one technology here that we've been using and in Cleveland was what we call MR fingerprinting. It's basically looking at T1 and T2 absolute values on the MRI. The interesting part of it here is maybe enabling us to see something that is epileptic on the MRI, if it can happen. So, like an example here, you see this patient with bilateral periventricular nodular heterotopia. Typically, we don't know, based on the MRI, which one is epileptic. We have to implant some of them. We cannot implant all of them. And if we're lucky to see, like in this case, that this is the epileptic one, and we do this MR fingerprint, and we measure the T1 and T2, we're able to differentiate. This is work by Irene Wong, who's leading our group for imaging, to basically show that the epileptic focus may have different absolute T1 and T2 values, in particular T1. Maybe telling us that in the future, we may be able, if this is validated, to see the epileptic area and maybe avoid doing SEEG on them and do and burn it like we can now using laser or maybe high-intensity focus ultrasound. Second thing here, we have to use less invasive approaches. We can do laser, for example, and here in India and many places in the world, doing more of the radiofrequency thermocoagulation with SEEG placement. But there is something interesting here that we are starting to use in the United States, is using high-intensity focus ultrasound to ablate deep foci in the brain without opening the brain, without putting even a probe inside the brain. And that is something giving us hope for cases like periventricular nodules to just do the ablation in these patients without opening the brain. Now we have to start to use AI, and everybody's talking about it. And AI will be very helpful if we know, or if we cataloged our data and make it available, because we need big data here in order to come up with better results for this. The lowest hanging fruit is using AI to better analyze the EEG. Post-processing or live detection of seizure. I just did a PubMed search 3 days ago on AI and epilepsy, and we found here 1,864 reports, PubMed. It's good. And the main challenge here is defining what we call the ground truth. AI is as good as we are in defining what our goal is and defining the data in this. In the midterm, I think here we have to pursue the molecular visualization of the lesion. But we cannot do it if we don't identify molecular targets. That's why genetic is going to be important. Let's show you here a sneak peek on something we can do. This is an 11 Tesla image of a cut of the cortex. A patient who had cortex dysplasia, this is small sulcus here, and imaging the NMDA 2B subunit. What we did here was this. We took that piece, we put it in an artificial CSF with oxygen. We based it in a GAD-tagged antibody against the NMDA2B receptor. And we incubated it for 30 minutes, washed it out, and overnight did the MR imaging with 11 Tesla magnets. It looks like it is an immunocytochemistry. Now, we need two things here. We need the target, which we may have some targets here and there, but we need something to cross the blood-brain barrier. That is, it would not cross. Now, can we open the blood-brain barrier? There may be some hope about this. So we need specific substrate. And that will be, hopefully, next 10 years. I always, 10 years ago, I said next 10 years. 20 years ago, I said 10 years. I still say 10 years to identify better targets. Now, why we may be luckier now? Because genetics is becoming very good. We're becoming good in genetics. And using our thousands of epilepsy surgery samples, we may be able to identify some novel genes that could be either targets for diagnosis, not the genes per se, maybe the protein that downstream here to be, and some of them may be target for treatment and monitoring the effect of treatment. It's a long way, but this is something that I think the train has left that station from gene therapy standpoint. This is an example of a paper that we published in Brain 2 years ago, looking in our patients with a group in Erlangen, Germany, Ingmar Blümke's group, looking at somatic mutation in various lesions. We have here FCD2, we have hemimegalencephaly, polymicrogyria, FCD1, MOGI, hippocampus cirrhosis, and the congenital tumors. Why is this important? Because these are somatic mutations. Any of them could be a target for gene therapy. And that is the hope that hopefully we're moving very soon into that field. This is here a study looking at multiple reports on gene sequencing, identifying 247 unique genes. If we look here, majority of them are enzymes, ion channels, signaling pathways, receptors, and then transcription factors and everything else. So we may have candidates coming up soon. If you look at the slide and look at slide from cancer, any type of cancer, 20 years ago, this is where they were. And we know where cancer is now. We don't need to reinvent the wheel. We can think about it like cancer did in the past. Something here, it's very interesting, is the field of epigenetics. It's basically normal DNA, but there is a chaperone, a chemical reaction, methylation over this DNA, making it like it is a mutation. The interesting thing about it here, it can be dislodged easy, but it has to be identified. This is a study we have not published yet. We submitted it recently, worked with Samuel Oster down in Melbourne, Australia. And what we're looking here is at a single gene level for a specific mutation in the tissue for DNA methylation. And what we found is there is a difference, clear difference between FCD2A and FCD2B and control tissue. In the tissue. But what's more exciting is that this change, exactly same, we're seeing leukocytes in the blood. So could we start to think about making a diagnosis of a pathology without taking a piece of that pathology, thus enabling us to use less invasive techniques to manage these patients? I think we may be getting there soon, and this data should be verified, obviously. In the United States, it's the first study for gene therapy for epilepsy started 3 months ago. And this is by a company called Unicure from the Netherlands. It's a French-Dutch company. They're doing here is a virus-tagged microRNA against the canic acid receptor, one of the glutamate receptors, for treating patients with mesial temporal lobe epilepsy. You can see here there is a probe inserted from occipital, injection of a total of 1.5 milliliter of virus-tagged microRNA against the kainic acid receptor. We have not enrolled patients yet. Hopefully within next year, we'll be enrolled. We need to enroll 12 patients total. And because this is a safety study with secondary endpoint actually to look for seizure freedom. Is it going to be earth-shattering from a result standpoint? I hope so. But I think the most important thing is a proof of concept. Like regulatory agencies like the FDA in the United States, they are approving those for treating patients with medically refractory epilepsy. Now, I think the bigger promise is imagine this, patients with focal cord dysplasia 2A or 2B, blood test knowing what gene, and knowing just going and doing a antisense oligonucleotide targeting that particular mutation, injecting it through the vein after brief opening of the blood-brain barrier using low-intensity focused ultrasound. It opens it for 1 hour. It's been done now for brain cancer treatment, chemotherapy. 1 hour opening the blood-brain barrier, injection in the vein of antisense oligonucleotides, and patient goes home. Will it be the future? I think it's closer than we think. Now, there is another trial actually here using stem cell therapy to engineered stem cells to become GABAergic interneurons in the United States for patients with mesial temporal lobe epilepsy due to hippocampus cirrhosis. Multiple centers in the United States, they started to enroll. I don't know exactly how many have been enrolled, but they need to enroll 12 patients like the gene therapy one. So it is another sneak peek in the future. This is a busy study here, a busy slide, and my final one is basically to imagine how the future, the near future, next 5 years, midterm, 10 years, and beyond 10 years, where we're heading. As I mentioned, I think at this point, we have to make surgery less invasive, smarter, and yielding better seizure outcome, better than the 55 or 60% that we have now. And this will be done better imaging, sophisticated post-processing, and using some AI tools in either analyzing the images, the EEG, and maybe start to see some use of engineered medications against some potential gene mutations, focal delivery of those. The second phase is going to be most likely here. We are getting more into the gene therapy sphere. At the same time, there is a work to be done on defining better targets from our gene sequencing to go downstream and identify the proteins. Hopefully, getting a blood test to know what we have, what lesion, and identify what we need to do later on. But the last frontier that has been escaping us since Penfield in 1950s, he talked about a lesion in the, when he described hippocampus cirrhosis. He talked about a patient who have a febrile convulsion in between the age of 1 and 5. Will have a seizure and no seizures now for 5, 6, 7 years. And the first part of the second decade, they have their seizure and they continue to have seizures more and more frequent. Hippocampus cirrhosis. But what he said here, there is a period of ripening of this, the lesion becoming ripe, epilepsy become ripe. And this is what we call now epileptogenesis. We know in animal model, we kindle, we stimulate that hippocampus once, twice, 3, 4, 5 times. We make this hippocampus epileptic. This is a process of epileptogenesis. If we are able to identify it better and the factors or biomarkers of this, we may be able to spot it early after the patient who had a head trauma or at-risk patients, and maybe deliver a medication before epilepsy happens. That should be our dream and should be the plan for generations to come. Thank you again for this opportunity.
Thank you, sir, for an excellent lecture. Now we know what we can tell our patients, what their expectations are in the management of epilepsy in the future. It is a great tribute for the centenary year of Hans Berger and to our Professor N H Wadia Oration, who are the role model for all of us. Thank you, sir, for accepting our invitation.
Thank you.
I request the chairpersons to facilitate the speaker.